So I'm Guillaume Butler-Laporte. I've been for the last 2 years now working in Brent Richards' lab. He's a genetic epidemiologist and a clinician scientist, and I am also a clinician scientist. I graduated from my residency in infectious disease and medical microbiology in June 2020 at McGill, and then I decided I wanted to continue in research. And I thought, what's interesting right now going on, and I had the mathematics background going back to undergrad, and I wanted to continue doing that. So I said, oh, genetics is interesting. And so out of nowhere, this pandemic hit just as I was starting to do some real research in genetics of infectious diseases. So to me, it's been very interesting to combine all these three interests: medicine, genetics, and mathematics, for the last 2 years. So that's what I've been doing.
That's amazing. Yeah, maybe something positive that comes out of the pandemic is the start of this work that you're doing. So can you tell us maybe a bit more about the current work that you're doing?
Yeah. So for the last 2 years, really in our lab, we pivoted all the way to the pandemics and trying to find genetic determinants of what causes COVID and what causes specifically severe COVID, because especially now with Omicron, we know like it's almost a given that eventually the vast majority of us are going to catch it through no fault of ours. But there's a small percentage, even in the first waves, that people got very sick from it, and we really didn't understand why. We knew that older people they were sicker, that generally immune suppressed patients would fare worse, and these all made sense. But why would a 30-year-old person die of COVID? And that was through our work essentially in the last 2 years and through everything we've done, we've been trying to piece the puzzle together. And so we've been taking active roles in two big projects. The first one is the BQC19, or Biobanque Québécoise de la COVID-19, which is a very exciting collaborative effort between many of the hospital centers who are prospectively enrolling patients with COVID and following them and genotyping them and doing all types of bloods. And we have a really rich set of information and database on every patient. It's very exciting, and we use that to then discover what made them predisposed to severe COVID. And so that's the first part. And these are led by Vincent Mousser at McGill, and Brent is part of this also. He's leading the Jewish General Hospital wing. And then on an international scale, we're part of the COVID-19 Host Genetics Initiative, which was started very early on in the pandemic. And essentially, it's just a consortium of a whole bunch of geneticists throughout the world. Mostly the leadership is mostly in Finland and at the Broad Institute, but we also made our way inside this leadership group. And essentially, it's just a bunch of geneticists. We're like, okay, so we're used to studying other diseases, but now we have this pandemic. What can we actually do to answer the same question? And so I believe the latest effort in terms of genomics that stemmed from this consortium is more than like 1.5 million participants. It's gigantic, just like gigantic. And we discovered, actually, like, surprisingly, and I guess that's going to answer one of your questions, the probably most surprising thing is that there are clear genetic determinants of COVID-19, and we weren't expecting to find them, at least not so clearly.
That's really incredible. And can you tell us maybe a little bit more about that then? Like what is the most surprising thing that you have found through this incredible international work?
Yeah. So, before the pandemic, genetics of infectious disease, of the host genetics, so the genetics of the people who catch the infection, there was, of course, a lot of work being done on that, but it was really the first time that we had the opportunity to study an infectious disease at such a large scale, because everyone was catching it and every health system and every country was suffering through it at the same time. And so it was, we like I don't want to say, sorry, that's my dog. uh, I don't want to say that we didn't think we were going to find something. It's just we were kind of in the dark as to how much we were going to find, because like there was already plenty of research done on cancer genetics, osteoporosis genetics, diabetes genetics, asthma genetics, all sorts of chronic disease. But acute disease like this, that you catch it, you get very ill, and then you recover or not, but hopefully you recover. We didn't really have sort of a research history on this. We didn't know where it was going, and we didn't know how many patients we need to enroll. We didn't know where the signals would be. We had kind of some idea, but it was really, we had never done this really. And so, I believe the latest result, we have found 20 locations across the genomes where clearly if you have a genetic variant as a patient in one of those areas, you're at much higher risk of developing severe COVID. And some of those variants, even if the effect may not be great, you're not going to quadruple your risk of severe disease, but because there are so many of them, and every single one of those variants are actually quite common. Like some of those variants, like 10 to 20% of the population carries them. So if you add them up all together, some people, I mean, that explains a large part of the puzzle as to why younger patients would fare worse despite having none of the usual clinical risk factors. So yeah, essentially keyword is like this, at least, but probably more than 20 separate locations in the genome where if you have something there, if you have a specific genetic variant there as a patient, statistically, you're going to do worse.
So as a non-geneticist, then my question is it 1 out of the 20, 2 out of 20? Does it make it worse when you have more of those variations? How does that work?
So it's not exactly an addition, but it almost is like that. So if you have 20 of those 20, I say 20, it's approximately 20, and then there probably are more that we haven't discovered, but there's a few dozen at least. If you have, like, the more of those variants you have, the worse statistically you're going to fare. It's always a game of chance, of probability, and of course with vaccination you decrease that chance quite a bit. But yeah, like we saw that clearly. One of the striking feature of COVID-19 is that there are very common genetic determinants of worse COVID. And they're so common, in fact, that we believe that it would explain a lot of the variation and a lot of the severe cases, especially in younger patients.
Go ahead.
No.
What I was just going to ask, what does that percentage look like then, especially for like young or healthier people? What percentage of those people have these variations?
So it depends. I'm going to give you an example of the ones that we probably know the more about. It's the specific locus, so a specific location on chromosome 3, which is the first genetic signal we saw on a large scale that affected severe COVID. And approximately 14% of the population has at least one genetic variant in this location. And if you have it, the risk that you develop severe disease is probably 2.5 times higher, 2 to 2.5 times higher than the general population. So for something that 14% of us carries, that's remarkable, right? Other variants are sometimes more common and sometimes less common and sometimes worse and sometimes not as bad, but it's kind of, everyone is kind of in that spectrum of how many of those variants do you have. But yeah, like the first one we found, it was striking. Like you double your risk of severe disease, and 14% of us carry that. So
is that now like common knowledge, would you say, amongst all of these different centers that are collaborating, sharing this information? Like, and is that reaching the clinical setting as well?
So good questions. The first part, yeah, I think every geneticist that studies COVID at this point knows about this one because it's been replicated time and time again, and everyone agrees that this is for sure real. Of course, anytime you look for associations with a disease across the genome, that means technically you're doing millions and millions of statistical tests. Just by chance, you expect some of them are going to be spurious associations that are wrong, and it's just like because you do so many, some of them are bound to show something, but that's not real. But this one has been replicated times and times again, so we know it's true. and yeah, everyone in the field knows at this point. The question about clinicians, which of course to me is important. So that's interesting because there's a few things you can do once you have genetic determinant of any given disease, but in this case, infections. Of course, you can, like this idea of adding up risks, you can actually devise a what's called a polygenic risk score or genetic risk score. There's multiple ways to do names to it, which essentially what you would do is once you devise this score, you can actually provide a probability to a patient. And something like COVID, it hasn't panned out just yet, probably because we haven't, one, it hasn't been a very long time. Two, up until recently, you probably didn't have the sample size to do this. And three, clinically, it's hard to apply this in a disease that hurts you so fast. So usually these polygenic risk score are very useful for something like breast cancer. They actually perform extremely well. And if a woman has a very high risk in these one of those scores, I mean, it's things like this that companies like 23andMe are starting to do. Okay, so it's going to come out clinically. In infectious disease, one of the great things it's been that genetics has been doing is actually to narrow down a list of targets that we can use to, well, inform clinical trials. And so, you know, I don't know how much you've been following the clinical trials, but because I'm a clinician, I've had to, and it's surprising. Like one of the treatments that we use on a regular basis, it's called tocilizumab, which is an interleukin-6 inhibitor. Well, you know, you can find a signal for interleukin-6 inhibition in one of those on one of those 20-ish loci. Another one is baricitinib, which is a TYK2 inhibitor or JAK1, but at TYK2, the gene for TYK2, we see a signal right there. You know, there's a signal at OAS1, which is a protein that essentially degrades the virus, essentially. And we have different molecules that we can actually target to increase the amount of OAS1. So this has been more the way that genetic determinants of infectious disease has been used currently. It's to inform clinical trials and to try to make sure that because clinical trials are very expensive and take a long time, so we want to, you know, get the most out of them, and we try to prioritize them, and that's exactly what's been happening. So there's currently, I'm not part of this trial, but there's a trial going on it's based out of the UK, but they have branches everywhere. It's the recovery trial, or the recovery collaborative group, and essentially they like one of the leaders there is actually one of the leaders in the COVID-19 host genetics initiative, and he's been very blunt about it. So the results we see in our genetic association studies, we transfer them right into our clinical trial pipeline. So that's what we've been seeing, and for infectious disease, this is where it's been helpful, at least for now. In the future, there's probably other avenues, but for now, that's what it's been useful for.
That's really amazing. That's really, really interesting. Thank you for sharing that. Is Canada doing any kind of clinical trials like that you're aware of at this point?
Yes, I mean, there are groups, again, so I'm not involved in clinical trials for COVID-19, so I can't tell you too much detail about it, but I know that recovery trial has branches in Canada, that's for sure. They're probably not difficult to find, but I know like there are hospitals who participate. I have, like at the MUHC, many of my colleagues that's where I did my residency. I know many of them are heavily involved in clinical trials. But the main one, from a genetics perspective, the one that has been the most genetically informed is the one based out of the UK. But yes, I think it does have a few branches in Canada and in other countries.
Very exciting. So I guess just shifting back then to genomics research, what do you think are some of the key lessons that will have come out of this work with COVID-19 that other genomics researchers can hopefully apply?
It's you know, if we want to get like the name of the game in genetics, I think, is replication, and it takes sample size, and we're starting now, and I hope that COVID-19 has really been a catalyst for that, but we're really starting to see massive large-scale genetics effort. And not only large scale, but also that include participants of different genetic ancestries. And I use the word genetic ancestries because I don't want to use specifically ethnicity or race and all that, because it has a very specific meaning, but it does share a bit. But before that, you know, in genetics, unfortunately, due to legal reasons, due to medical reasons, due to just like demographics, the European ancestry individuals really just oversampled and overstudied, and we probably missed a lot of great genetic stories by not studying the other one. But COVID-19 is also a bit of a catalyst for this. So I guess the big takeaway is we need to just like learn to work together, because when we do, we achieve great things, really. Like,
so you are seeing then improvements in how studies are being conducted now and how that research is being conducted now in terms of the diversity?
yes, well, I mean, not only that, we just, one of our papers just came out today in Nature Genetics, and essentially what it shows is at this specific OAS1 gene location where we saw a signal, and we couldn't actually understand where the signal was coming from. There were a few, like, there were actually a couple of genes in the vicinity that could explain this, and there were a couple genetic variants that could explain this. But this is because the signal was too noisy in European ancestry. And then if you just took the time to study African ancestry individual, the signal became like pristine clear, and we knew exactly what was the mechanism of genetics. And I'm not always saying that it goes in this direction. I'm sure there's sometimes an African ancestry population that's a bit muddier, and then you go and European and it's clearer. But the trick is like, when you in genetics, what's clear is that when you study different genetic ancestries and increase the diversity, it's not only, you know, everyone gains, all of these genetic ancestries gain something out of it. And that's become clear. So yeah, I mean, I just published on this with others today on this, so I'm pretty happy. But yeah, I think it's something that, I think COVID has been a catalyst for that. Everybody knew about it again already, but it's definitely been a catalyst for just larger, more massive, and more diverse sample sizes.
That's great. That's really exciting, and congratulations on your publication. We'll definitely share that around with this video as well in the interview. So thank you for sharing that. Is there anything else from that you'd like to share? Any other interesting outcomes from that work?
Oh, well, I mean, essentially what we did is there was a specific locus, a gene called OAS1, as I said, whose role is to produce a protein, OAS1, that really kills the virus. Essentially, it's a bit more complicated, but that's more or less what it does. And we didn't know how it did it. Specifically, every gene is able to encode for different proteins. Really, when the gene is transcribed to RNA, and then the RNA is spliced differently, so in different, so then you have different types of RNA segments who are then translated to proteins. And so from one gene you can have different proteins that can actually be produced. And so what we found was that there's a very specific, now I'm blanking on the word, but yeah, there's a specific isoform, so a specific version of that protein in OAS1, and it's this specific version that is actually helpful against COVID-19. Other versions are helpful probably against other viruses, but this specific version is helpful against this. And so from a pharmacologic perspective, it's important to know which one we want to target, because if we target the wrong one, well, it's pointless. So because we started with a genetic signal that was mainly stemming from European ancestry individuals, and then because we then decided, oh, we're going to look at Africans because their genetic architecture, that locus is different, maybe they're going to be able to really help us determine what's going on and which one of those isoform is helpful for COVID, and that's exactly what happened. So yeah.
Wow, that's definitely over my head, but sounds very, very interesting. So thank you for sharing that.
Yeah, I tried to
Yeah,
vulgarize it, but I'm
no,
sorry.
you did. You did a good job of explaining it. So thank you for doing that. So I mean, this is all really exciting. Is there anything, I guess, in terms of just genomics, maybe in the future, where do you think that we're going from here? Like, there's a lot going on, and it's really incredible. And what do you think will be next in terms of hopefully once this pandemic is over?
You know, I'm as a clinician, I'm very excited as to, you know, where it's going to, how it's going to affect our clinical work. And you know, I might be too young, but I remember when I was a teenager and I was reading like scientific magazines, and I was like, oh, the Human Genome Project is going to change the way we treat patients, and it's true. It hasn't really panned out, but I think it's because, well, we weren't there yet. There was a lot of homework that needed to be done, and also maybe our expectations weren't the right ones, and maybe we went around it the wrong way. But now that, you know, 20 years has spanned, now I'm in the middle of it. I don't just read about it. I'm in the middle of it, and I can actually see where it's going. Okay, so like for some chronic diseases, those polygenic risk score, they're going to come in the clinical practice, and if they don't come in clinical practice, they're going to at least come massively in clinical trial design, and that's going to impact us all. Maybe they're also going to come in clinical practice too, for sure. But the other way that's going to come is, you know, with all these massive large scale studies, it's clear, and big pharma, quote unquote, has picked up on it and has invested millions of dollars into using these genetic signals to inform drug development. So it's funny because 20 years ago, we thought, you know, like the Human Genome Project, the next step was going to be like everyone gets their genetic sequence, and now we know exactly what all your diseases are. Well, a bit more complicated, but still, in a few years, I believe it's going to be everywhere. Maybe not at, you know, at the interface of, you know, the patient and the physician, but everything we will have learned will influence this relationship. So maybe a bit more indirectly, at least for now, but definitely it's going to be pervasive, I think, by the end of my career. It's going to be everywhere.
That's great. It's very exciting. And it's an exciting field to be in. So is there any final thoughts or anything else you'd like to share, maybe for other young researchers that are interested in this field? Maybe, you know, they're just starting out or in the same stage as you, but any advice that you have maybe for people, especially Canadians that are, you know, interested in this?
No advices. You know, what I've learned is everyone is eager to help everyone in genetics. Coming from medicine, I can say maybe tongue-in-cheek that it wasn't always the case there. But in genetics, I found that everyone's always there for each other. So don't hesitate to look for help, and don't hesitate to involve yourself in different research projects. There's so much to learn, but there's also so much to contribute to. You know, I'm a clinician. I do have a math background, but I'm not a pure mathematician, and I work with people who clearly they're very deep into mathematics, and then I work with people who are more epidemiologists, and then I work with people who are more who come from the biochemistry and biology field, and genetics is just like kind of this, it's in the middle of all of this, and it's very exciting. But that means that there's always someone in the room that knows more about something than you, but you may know more about something than that person about something else. So you have to talk to each other, and you have to learn to, learn from everyone, because it's the only way we can advance, really. And it's great. It's great. but yeah, that's my advice is, you know, try to touch a bit of everything, because that's very helpful. But learn to work with others.
That's great. Very good advice. Thank you. And any last thoughts? Anything else?
No, I think that's it.
Okay. Well, I really appreciate your time again, and thank you for sharing all these insights, and also the incredible work that you're currently involved with is very exciting. So thank you for taking the time to share that with everyone who's listening and watching. And we're excited to see more coming out of your work. So thank you.
Thank you so much.
Take care.
Bye-bye.